Hi guys, welcome to part 2 of the class. So we finally finished part 1. You guys took your first midterm, and now we're moving on to part 2. So for the first part of part 2, then we're going to talk about amino acid neurotransmitters. Specifically, today's lecture is going to be on glutamate. Now, before we get into glutamate specifically, let's just talk about what is an amino acid neurotransmitter. Now, these neurotransmitters are what we call non-essential amino acids, and that's because we don't need to get things from our diet in order to make these amino acids. So these are amino acids where the body kind of makes it itself. So this is what makes it distinct from those monoamines. Because remember, with monoamines, the tryptophan is an essential amino acid that we have to get from our dietary sources. And then the tryptophan will then go on to make our monoamines. Whereas with these amino acid neurotransmitters, then they're non-essential. Now, amino acid neurotransmitters obviously are amino acids, so you'll want to know the basic structure of an amino acid, that there's an amino group, a carboxylic acid group, and then some kind of side group or an R group. Okay, so you want to know what these three kind of side things are. You don't need to memorize every single amino acid that there is, but you will need to know the two main ones we're going to talk about. So today, glutamate, what does glutamate look like, and then GABA, which we'll talk about after reading weight. Now, our amino acid neurotransmitters can be broken down into two functional groups. So the first functional group would be excitatory amino acid neurotransmitters. So the big one here is glutamate. That's the one we're going to focus on, but it also includes aspartate and some others. And then there's inhibitory amino acid neurotransmitters, where the big one's going to be GABA, but it also includes glycine. So know what makes the amino acid neurotransmitters different than our monoamines, and then how do we break up our functional groups. So here's just showing the two most common excitatory amino acid neurotransmitters and the two most common inhibitory. Now, most or almost all amino acids are proteinogenic. In other words, they're used to create proteins. And of course, these amino acids are encoded by DNA, and then they go on to make proteins. Now, the only exception to this is GABA itself. So GABA is not proteinogenic. It's a specialized amino acid that is really only used as a neurotransmitter. And so there aren't proteins made up of GABA. It's the only one. But you will need to know what proteinogenic means and know that all the other ones are proteinogenic. So if we're looking at all of these neurotransmitters, then we can see that we've got our groups that signify them as being an amino acid. And then we've got our R groups that's circled in the dotted red, which is what makes them different from each other. Now, if you're looking at GABA, you'll notice GABA is a little different. It does still have both the amino acid group, or the amino group, and the carboxylic acid group, but that carboxylic acid group is contained within the R group. But it is still an amino acid because it does still have these two groups. But it's because of this kind of weird structure that makes it not proteinogenic. Now, if we're looking at just the excitatory amino acid neurotransmitters, which we'll focus on the rest of this class and the next one, then again, the two most common ones would be aspartate and glutamate. So aspartate is the second most popular one. It's released in a calcium-dependent manner. It's not stored in vesicles, which makes it kind of unique. And it can be directly released from the cell cytoplasm. So it's really not a classic neurotransmitter that we talk about a lot, where it's stored in vesicles and released via vesicles into the synapse and all that kind of stuff. So it's a bit unique. And aspartate tends to act on glutamatergic receptors. So there aren't like asparatergic receptors or whatever. They don't have their own receptors. They act on the glutamate receptors. We also don't know a lot about aspartate, so we don't really know what the physiological functions of aspartate are. And because we don't know a whole lot about it, we're not going to cover it much in this class, but I do want you guys to be aware that it's there, and that it's also acting on those glutamatergic receptors. Now, for the rest of the class, we're going to focus on glutamate. So glutamate is the most widely used excitatory neurotransmitter. So of all the neurons in your body, about 90% of all neurons are glutamatergic neurons. And about 80 to 90% of the synapses are glutamatergic. So that number differs obviously because you can have like a glutamate to glutamate thing, or you can have like glutamate to dopaminergic neuron or synapses, whatever. But obviously glutamate is like huge, right? And so glutamate's great because it is excitatory and it also mediates as a very fast excitatory neurotransmission. And we'll talk about just how fast later, but it's like real quick. And it's involved in pretty much like everything in the brain. So from sensory to motor coordination, emotion, cognition, memory, retrieval, like it does it all. Again, it is a proteinogenic amino acid. We see it throughout the cell, so it's not just in the dendrites, in the axon terminals to like send the neurotransmitter. We see it in the dendrites, in the soma, in the axon, as well as in those axon terminals. They do tend to be concentrated in the presynaptic compartments, so we don't see them too much like right at that postsynaptic compartment, so like right at the postsynaptic membrane of the postsynaptic cell. They kind of stay a little bit away from that the synapse in the postsynaptic side, but they are found throughout the entire cell. So how do we synthesize glutamate? It's going to be pretty simple here. We've got glutamine. Glutamine, then we use glutaminase, and it turns into glutamate. It's that simple, guys. So glutamine uses glutaminase to turn into glutamate. Amazing. When we start talking about how it's broken down, you're going to see it's pretty much the exact opposite. So glutamate is made from recycled glutamine. So we'll see how in the reuptake, glutamate will be broken down back into glutamine. So it's kind of just this never-ending cycle of glutamine to glutamate back to glutamine, and so on and so forth. Now, once glutamate is made, then it's packaged into our vesicles so that it can be kept separate from other neurotransmitters that may be made in the same cell. or just to keep it separate from everything else. Now, remember that our monoamines would get put into vesicles using VMAT. Glutamate gets put into vesicles using V-GLUT. It's the vesicular glutamate transporter. It's amazing how it's all pretty much the same. Now, V-GLUT, there are 3 different kind of types of V-GLUT, and the type of V-GLUT used kind of just depends on what part of the brain you're in, but it all kind of functions the same, where it takes the newly synthesized glutamate, puts it in a vesicle so it can later be released into the synapse. So when talking about our different types of V-GLUT, we got V-GLUT 1 and 2 do pretty much the same thing, and they're expressed pretty much distinctly in those glutamatergic populations. So in a glutamatergic neuron, then you're going to find V-GLUT 1 and V-GLUT 2, depending on what part of the brain. So you can just see here, you don't need to memorize which parts of the brain, but it's just showing the dark colors is V-GLUT 2 here, and then over here V-GLUT 1 is in these parts of the brain. So the cortex is using V-GLUT 1, the kind of midbrain is using V-GLUT 2 more. Again, you don't need to memorize the parts of the brain where V-GLUT 1 and 2 are expressed. Just know in the central nervous system, glutamatergic neurons have V-GLUT 1 and 2. We also have a V-GLUT 3, which is kind of unique because we actually find it in non-glutamatergic neurons. So we find V-GLUT 3 in GABAergic, cholinergic, and monoaminergic neurons, which suggests that there may be some kind of modulatory function of V-GLUT 3 in these other types of neurons. Now, in the hippocampus, then we actually see expression of both V-GLUT 1 and V-GLUT 2. And in the hippocampus, then depending on which V-GLUT is the neurotransmitter is packaged into, actually determines the release probability for that neurotransmitter. So pretty much what I'm saying is, so here in A, this is just a cross-section of the hippocampus. Anything labeled red expresses V-GLUT 1. In picture B, the same thing, but anything in red is expressing V-GLUT 2. So it's the exact same area of the brain, just pictures taken for 2 different V-GLUTs. But because both are present here, then kind of how does the body or the cells decide which vesicle to load our glutamate into? And it kind of depends on what we want the release probability to be. So if glutamate's packaged for V-GLUT 2, then it increases the probability that the vesicle will then go and be released into the synapse. If it's packaged into V-GLUT 1, it actually decreases the probability of the neurotransmitter being released into the synapse. So V-GLUT 2 has a higher probability of binding with the synapse, whereas V-GLUT 1 has a lower probability. Now that doesn't mean V-GLUT 1's never going to bind, it just means it doesn't bind as often. Okay? Now also, just a note that when you release glutamate into the synapse about 10% of the time, then you'll also be releasing zinc. And that's just because zinc can also be transported into those vesicles using V-GLUT 1 and 2. So just wanted to note that.